Hello, I'm Lisa Back, and I am going to be bringing you Saladin's Anatomy and Physiology. It is an 8-credit course broken into two semesters, A&P 1 and A&P 2. In this first introduction semester, we will be going through chapters 1 through 16 through the nervous system, and then in the second part we will be picking up with the endocrine system and going through reproduction and then aging and senescence. So I'm really excited to get started with you all this morning, so we'll jump right in, okay? The introduction to anatomy and physiology would, as you would maybe expect, define anatomy and physiology for you. So I'm going to need for you all to know the difference between those terms. And anatomy is actually the study of form and also where structures are actually located. So when we study anatomy, it makes sense that we study it along with the physiology, which is defined as how those structures function or work. So it really makes very good sense that we pay attention to the structure, which will always be dictating the function. This is why our ears look, their form is different than the structures of our nose, our nares, because they have very different functions, don't they? So if we pay attention to what something looks like and where it's located, it should help us to understand how it is actually functioning at that site. And this isn't just with things that we can see with our naked eyes, but it is also even at a microscopic level. Structure and form will always dictate how something is working, so its function. Again, anatomy is the form, the structure, and where things are located. So all of that together is anatomy. And then using those, it can help us to understand how something is actually working, or the function of it, which is physiology. So we are going to look at a lot of different ways that we can actually learn anatomy, and then different ways we can think about the physiology. So in examining form and structure, we certainly want to inspect, so we want to pay attention to what we can actually see. So we definitely do detailed inspections, but we also sometimes are going to touch. So palpation means to touch. Sometimes we're going to need to listen. Auscultation means listening. Percussion is actually a combination of palpation and auscultation. In percussion, we're actually sometimes tapping on structures and listening for echoes to see if maybe an organ is filled with fluid or maybe it is actually hollow. So percussion is kind of a combination of both palpation and auscultation. One of the best ways to learn anatomy and what we can see with the naked eye, which is actually defined as gross anatomy. Gross anatomy is anything that you can see with the naked eye, is to actually be using the human body. So cadaver dissections, where we use a dead body to actually cut up and dissect and to look at different body parts, is that human cadavers are the best way we can learn human anatomy. And then again, we're using anatomy to help us to understand physiology. So combining those two, using the human cadaver is by far one of the best ways that we can study human anatomy and physiology. Also, looking at anatomy on living specimens as well. There is something called comparative anatomy, and in comparative anatomy, we're looking at other animal species. So we can, use other mammals, if you will, to help us that have very similar organs to help us to understand human anatomy and how it actually works. So you have with this series available to you something called the Revealed Anatomy and Physiology. So that link will take you to a cadaver series that you can use your mouse, and you can actually go from the most superficial structures like the integumentary system, the skin, and cut away and see the superficial muscles, continuing to cut to see the deeper muscles, and continuing on down until you are deeper and you can actually see some of the organ systems all the way to the skeletal system. So the beautiful part about this Reveal series, where they used human cadavers, an adult and a child, is that you can use these over and over again. So when you want to go back and see something, you can just start at the beginning. In an actual anatomy lab where you have the actual cadaver, you get one chance at it. So there's this one opportunity with the dissection to actually commit to your knowledge centers these structures, and after that you really can't put them back. But in this series you are able to. Another beautiful thing about this series that's available to you is that these cadavers were recent cadavers, and so they had not been exposed to the preservatives that cadavers typically are in a medical anatomy lab. When a cadaver, well, once a person dies, the tissues immediately start to change, immediately. So to prevent too much of a change, they will take a cadaver and usually expose them to preservatives, formaldehyde and other types of preservatives that can slightly change the tissue consistency. So again, another real plus with this series, this simulated series that you have available to you, is that these cadavers were not exposed to those types of chemicals. So they are very much like they would have been in life. And also the dissections were just beautiful. You can imagine how difficult it is when you are removing the superficial skin, how difficult it is not to tear or cut the superficial muscles. But they weren't in these, and they are absolutely beautiful examples of how we can see the anatomy on this cadaver series. So that is definitely a great way to study anatomy, which helps us with physiology. It used to be that exploratory surgeries were done quite frequently. They're not done as much anymore because now we do have non-invasive medical imaging where we can actually do CAT scans and MRIs that can actually give us 3D imaging without having to actually surgically open up and cut and cause trauma to the tissues. So these are ways that we're still using to help us with diagnosis of disease. And in that respect, I do want to let you know that when we think about diagnosis of disease and in the health profession, that's really sort of how we're gearing, our study. We do know now, just as important in the medical profession and health profession, is that we know that we should be preventing disease. So while we have come a long way in diagnostic medicine, we have also, that has allowed for us to understand how things actually occur. So much of our focus now should be, the pendulum is swinging back, and we should be trying to help people prevent the diseases. Many of them are preventable diseases. So I want to talk to you about how we get to diagnosis. How we get to diagnosis is by using signs and symptoms of disease that help us to come to a presumptive diagnosis. So when we think about a sign of disease versus a symptom of disease, you should know the distinction and the difference between the two. A sign of disease is actually something you can measure and you can see and you can document that you have measured and you have seen it. So for example, a sign of hypertension is taking a blood pressure and seeing that the blood pressure results are what they are and documenting it. A sign of potential infection is fever, and you can measure someone's temperature, can't you? And if it's elevated and if it's normal or low, you can document it. A sign that someone may be having a dermatological event is a rash, and you can actually see the rash, you can measure it, you can describe it, and that is a sign of disease. So anything that you could measure or that you could see is a sign of disease. Just as important are symptoms, though, And symptoms are subjective. So they're the things that the patients are telling you that they are experiencing, but you can't really, they're subjective and you really can't see them. So a patient may say, I'm having headaches, or often I am feeling nauseous, or I have bouts of dizziness. Those are all very important things to be documenting, and things that will help you to get to what is called a presumptive diagnosis. So when someone comes in, you take the signs you can measure, you listen and document the symptoms you're being told, and hopefully that is narrowing down to what you think might be going on, which is referred to as a presumptive diagnosis. You're presuming based on signs and symptoms. To get to a definitive diagnosis, which means the answer, you would have to use those signs and symptoms and your presumptive diagnosis to figure out which tests, laboratory tests, need to be done that can give you the answer. The reason that definitive diagnosis is so important is because then you will know how to treat effectively. So that was some information that you definitely need to be aware of, and you need to be able to discuss all of what I just talked about. But that's what's very important in medicine, is that we know what we're dealing with as soon as possible, sooner than later, and that then we will know how to effectively treat. There is no such thing as a completely safe treatment. So once again, this should tell you, when I say that, it should tell you that it is so important that we get to that right, correct answer. Because no matter what we use to treat, there's always going to be some side effects from the treatment. So you certainly don't want to be mistreating someone. So this is, again, our biggest challenge in medicine is getting to that definitive diagnosis and then making sure that we're doing the very best that we can do for the patients and their potential outcome. Also, though, this should be maybe making you realize that what we really try to do, since all treatments have some negative effects, where we really want to be going to, especially in the future of medicine, is preventative medicine. Okay, so I added a few little things there for you, but I hope you stayed up with that and took those notes. So we are able to, with non-invasive imaging, trying to cause no harm, any more harm than we need to, we can use these non-invasive procedures like x-rays, CAT scans, MRIs, PET scans. We can use those to try to get to our definitive diagnosis. Still, sometimes exploratory surgery is needed. I think eventually that will be a thing of the past, and I hope that's sooner than later, because obviously anytime we cut someone open we're actually causing trauma. But sometimes it is still needed and required. Gross anatomy, I had defined to you, is anything that you can see with the naked eye. Cytology, if you ever see the ology, you know that this means the study of. Cyte is a prefix that means cell. It's going to be very important in our study here as we go through these 8 credits of Anatomy and Physiology 1 and 2, that when we first get introduced to a prefix, a root word, or a suffix, that you learn them. Because what you're going to find out eventually is that there's no such thing as a big word. I know that in medicine, much of the most difficult part is the terminology. But what you're going to realize is that as you go along and you have learned prefixes, root words, and suffixes, there's no such thing as a really intimidating big word. It's just a diagnosis, a word that you can break down, and if you do know the prefixes, root words, and suffixes, you can figure out what's going on. So that is going to be your challenge, is that anytime you hear a new prefix, root word, or suffix, that you learn them. You learn them now as we go. So cytology is the study of cells, and we're going to learn a lot about cells. Histology is going to be the study of, really, I know it says cells right here, but I would actually rather you know that it is the microscopic study of tissues, which we are going to realize are made up of cells. So to see a human cell, you do need microscopes, and you need the aid of those magnifying lenses to be able to see cells. They're not visible with the naked eye, so you can't see them on gross anatomy. But all of our tissues are made of cells. So to be able to see how those cells are working together to form those tissues, it's going to be vital that we understand histology. What you all are going to hear from me repeatedly is that structure and function, structure and form dictate function. So when we think about disease, which means that the person is no longer in a state of health or well-being, all of that originates at the cellular level. All disease is at a molecular, which make up cells, cellular level. All disease. So if it is happening at that level, then the treatment has to be geared at that level. So we spend a lot of time with cytology. We have an entire chapter that is delegated to cells. It's chapter 3. And we also spend a lot of time looking at molecules, which are made of the smallest things that we know, which are called elements. And how those elements come together and play or not is called chemistry. And that's chapter 2. So we will start there because there's no way to understand the human anatomy and physiology unless you know the molecular and cellular processes that are happening that make up our tissues. So this is just a little bit of terminology on the slide, but important that you know it. Now, a histologist, which is someone who has specialized in being able to diagnose disease at this level, is going to be looking for pathology at this level. So histopathology is actually studying the disease at this tissue level. I broke that down, didn't I? That word down. And let me just give you this word pathology all by itself, because you've heard of pathology. Pathology is the study of disease. So pathos means dark, evil, in medicine it means disease. So the study of disease, and histopathology is looking at it at that tissue level. Tissues are made up of cells. So before we go any further, I've already added a little bit for you, haven't I? Talking about signs and symptoms and the difference between the two, and giving you the difference between presumptive diagnosis and definitive diagnosis. But now I actually want to it's reminded me to maybe ask you about human anatomy and physiology just in its most basic introduction, that this is a science, right? So this is actually a science that certainly is our core science in the healthcare field, whether you are going into physical therapy or radiology or nursing or physician assistant, no matter what you're going into, this is the foundation. This class is the foundation of that. And I can promise you from my many years of teaching, but also my experience as a healthcare professional in clinical microbiology, as a clinical microbiologist, and pathology for years, that this is the foundation of the professional that you will become. So this is not a class that you want to take and just check it off your box that you got it done so you can go to the next level. This is not that class. This is a class that you're going to want to own the information. That means that your challenge is that you would be able to learn this material well enough that you could teach it to someone. This is not a crossing a T, dotting an I, checking a box class. This is absolutely your foundation of your profession. And I can promise you from, again, my 15 years in clinical medicine, that it is very obvious when someone doesn't have this foundation that is already somehow gotten out there in the clinical field. You don't want to be that person. You want to be having a very strong foundation with this. So that's my challenge to you. Also, the challenge is that sometimes the way that you learn is that most, and this is most of the students I've encountered, they come to me and they have K through 12, or for whatever reasons, they've learned by memorizing. You haven't learned if you've memorized anything. Because if you have memorized information, it is rare that you can go from something that you have memorized to critically thinking about information. When you have actually learned information, you can use it to critically think and deductively reason intuitively about the next step. And you will be able to make connections and be able to, and you know, I don't want to call it having common sense types of foundations in your knowledge, but because to be honest with you, it's not really that common, but you would be able to use it for critical thinking. And to do that, it means that while you're studying this information, you are not memorizing. I've had students say, well, I didn't have it that way in my notes because I had written it down this way. No, I do want you to take very good notes. But that's not learning if you've done it that way. When you are taking notes and then you are studying those notes, you should challenge yourself to think about how that you would paraphrase that information, how you would turn it upside down and be able to discuss it backward, forward, connect it to things that you already know. Be able to explain it, as I said, in your own words, paraphrasing it. As we go through this course, you're going to have lots of testing opportunities, and they are not for me. They're for you. They're for you to figure out how well you had a grasp of the information. But you are going to have that opportunity to know from your assessments how well you may have gotten that information, and they are for you. They are not for me, right? So that is going to be the challenge for you for this course, okay? I also want to talk to you a little bit about this as a discipline, as a science in the health profession. I have a lot of students who, when I ask them what is science, they have a hard time answering that question. What does the term, it's an umbrella term, science, right? But I have a lot of students have a hard time answering it. But I want you all to know that science is actually a way, a logical way to answer questions about the natural world. So that's what science, you know, in a nutshell is that it is a way that we go about answering questions about phenomena that are occurring in our natural world. And it is limited to the natural world. Now, under science, there are many disciplines, right? Subdisciplines under science. There's physics, there's chemistry, there's biology, there's mathematics. So we call those true sciences, and that's what I'm dealing with, what I'm talking to you about right now. There are social sciences that are a little bit different, but when I'm talking about actual natural sciences, that there are all those subdisciplines under that. Now, under biology, bio is life, and ology is the study of life. There are many subdisciplines under biology. We could study plants, which would be biology, which would be botany. We could be, plants are alive, aren't they? Zoology is the study of animals, right? But we have a very narrow discipline that we're going to be looking at under biology, which is the human anatomy and physiology. So I do want you to sort of understand what science is, and that biology, and then even further down, human anatomy and physiology is a science. So we have to go about in a very organized approach and process of answering questions about how the human body actually functions. So that's where we are. All right. I am going to define for you later homeostasis, but right now, maybe you can just jot down that homeostasis is a state of health, a state of well-being. That's what it's defined as. So we'll get to that in just a little bit. Again, we've been given the difference between anatomy versus what I said, physiology. So in physiology, this is the study of function or how something is working, right? This is actually anatomy. If you think about anatomy, and that anatomy are studying parts, right? So when did you start to study anatomy? Anatomy is actually really kind of easy because we started studying this before we could even speak. Before you could even form words that were understandable, you would have someone tell you to pat your head or clap your hands. And you could do that. Or you'd have someone, you're just a baby, and you'd have someone say, stick out your tongue. And you would stick out your little tongues, right? Because you had a word to that structure. And you were already learning, to touch your nose, right? So anatomy, if you will, is actually a very sort of easy study, science of this, because we've been doing this for a very long time. And we're going to continue in this class, certainly, and we're going to learn what are the accepted medical terms for structures, so we don't really call your head a bean, do you? Or what? We have all these slang terms for body parts. So we're going to get away from the slang terms and we're going to learn the actual terms for these structures, but it should not be difficult. I'm not lessening how you all will have to set. I'm not trying to make light of it. I understand that you will be learning a lot of bones and you'll be learning a lot of muscles and that sort of thing. So I'm not trying to make light of it, but the fact is it's not difficult. It does require time, though. So the time you have to be able to manage and be able to put in with anatomy. The labs that you have in these courses are going to almost exclusively be anatomy, which means that you will be labeling parts and you'll need to know where those parts are located in an organism, in a human being. But it should not be hard. Now, physiology, admittedly, is more difficult. It's going to be a little bit more difficult, and so that is where you're going to be spending the majority of your time. So the lab part is anatomy. It's about 25% of this class, but most of what we're talking about in this course is going to be physiology and how those parts function, how they're actually functioning, and what it's going to take for them to function in a state of health or well-being or homeostasis. So there are certainly subdisciplines of physiology. So neurophysiology, obviously the nervous system, endocrinology, hormones, the endocrine system, pathophysiology, mechanisms of disease. So my specialty when I finished undergrad was pathology, and specifically microbiology, which is the study of infectious diseases. So we do have lots of subspecialties within medicine, and that is a wonderful thing since there's been such an explosion in understanding human anatomy and physiology just in the last few decades, and so much information is coming out, new information every day. So this is a wonderful, wonderful thing, but there is a catch-22, and that catch-22 is the idea that sometimes when you get sent to a specialist, they're just looking at you in that narrow little view and not as a whole organism, not as a whole entity or being, but they're just seeing you in their little subspecialty, which is wonderful because they do have the most expertise about that. But it is going to be very important that you as health practitioners make sure that, and nurses, by the way, I consider to be health practitioners. They're the primary care providers. Nurses are the ones that are. Yeah, you can think about doctors as being primary care providers that are sort of managing things, but it is the nurses that are the hands-on. It is the nurses that are documenting most of those signs and symptoms. It is the nurses that are executing by doing the IVs, giving the meds, taking the care of. It is the nurses that are the primary care providers. I'm humbled to be just a small part of you guys that are going into nursing or really any of the health sciences, but I'm humbled to be a part of your journey here. And I do want to impress upon you how important it is that you really do get this very strong foundation, because sometimes you're going to have to be thinking on your feet and you only have seconds. Sometimes minutes are luxuries. So you are going to have to be, having a very strong foundation for this. If you don't believe me, think about this even just during this pandemic, COVID pandemic, and all the things that have gone on with our, medical professionals, our frontliners, if you will. If there had not been nurses in those hospitals, if there'd only been, let's say, the doctors, they would have shut the doors. The doors would have been shut because they wouldn't have been able to run it. Okay, so I'm here for you to try to give you the strongest foundation I can possibly give you at this point, and impress upon you that you'll feel like you've learned this if you feel like you could teach it. So that's your challenge. There are sub-disciplines. We do use comparative physiology when we're actually studying human anatomy and physiology. We look at other species as well that have very similar types of structures. We use everything we can to study human anatomy and physiology. Comparative physiology is also the basis for new drugs and medical procedures. So, a lot of people get upset, sometimes can get upset about animal testing, but, it is a way that we have learned a lot before we would take it to the human trials, because I think most of us could agree that, we don't want to be using humans as the guinea pigs for much of this, this new information that we have. As far as the history of how we have gotten to where we are right now, this information truly is sort of FYI. If I go through these slides and I skip something or just go over it in a very superficial way, you're not responsible for it. Or as far as testing goes, I think it's all important, but we have a limited, finite amount of time in this course together. So sometimes I'll say something's FYI, and it means you won't be tested on it. There are some things I will pull out though and say you should know. Like, it is interesting and FYI that over 3,000 years ago in Mesopotamia, they were doing the craziest things. How crazy? Herbal salts and, laying hands on and those types of things. Is that crazy? No, because I don't like the word crazy either. I wish I hadn't used that. But no, that's actually kind of all coming back, isn't it? Because actually, yeah, even 3,000 years ago, humans knew that actually preventative medicine and supportive care medicine, it was helping to heal. So we actually got away from that for a while because we were, diagnostic medicine at a molecular level was exploding, and we were learning so much that it became all about diagnosing disease, and we kind of got away from that preventative care of disease, supportive care, but it's kind of coming back, and that's really a very good thing. I do want you to know about Hippocrates. Hippocrates is considered to be the father of medicine, and I think most of you have heard of the Hippocratic Oath. So obviously we got the Hippocratic Oath from Hippocrates, and it pretty much says that if anyone is going to make a profession in the healthcare field, that you will do no harm. You actually take an oath that you will do no harm. And, this really is important. As important, again, kind of FYI here, Hippocrates and Aristotle and some of the earlier people that we learned so much from said that there was always going to be a cause and effect for disease, and that we needed to seek out the cause and effect of situations. This was kind of revolutionary at the time, though, because, and actually not just at the time, there's still many, many people who believe there are like these supernatural forces that cause disease, or that someone deserved it, or that there was some sort of whatever as far as disease. Just not true. So we do know there's cause and effect. There are things there are many diseases that are preventable, and that we could just, with behavioral changes, actually definitely affect the outcome, because there is cause and effect. And they really were, Hippocrates and Aristotle really were some of the first that were saying that we should really seek out identifying causes and effects of disease, and helping to diagnose disease in that particular way. So I am going to go past, which means I'm not going to ask you questions of a lot of the earlier contributors to where we've gotten to today. It's fascinating. If you can ever take a medical anthropology course, I would advise it. It's very fascinating to know the history of how we've gotten to where we are today. Some of the things that have been done would really truly blow your mind. So if you ever get a chance to spend some time with that, then I would suggest it. But we're going to continue on, and I do want you to know that something about the cell theory. This is a scientific theory, and I want you to write that down. The cell theory is a scientific theory, because you're going to have to explain to me what is meant by a scientific theory. Okay, you might think you know what it is, but you might be surprised. Okay, but before I actually tell you what the cell theory states and the premises of it, I actually want to jump down to another slide here that I think is important, and probably is important to do first. So as I said, an organism, an individual, an entire organism, an individual will be coming to you as health professionals with their signs and their symptoms of disease. Hopefully they'll be coming to you before then. They'll be doing preventative medicine care, doing annual workups and annual exams, and trying to use behavior and diet and exercise and all those things that we do that we know that can prevent disease. But everybody gets sick sometimes, and eventually, and aging is a natural process of the organism, right? And death is a natural, step too. So that's assured as well. So the organism. Now you might think, let's start here, because that's going to be the most fun, is to start with the organism. But we really can't, because we are made up of our parts, right? So an organism, if you think about this hierarchy, and I really need you to know this forward and backward, an organism is composed of their organ systems. So you knew that. You knew that there are organ systems, like you're seeing here an example of the digestive system. There's also, in this organism, there is the nervous system. There's the integument system, which is the skin, right? So there are many different organ systems. So this is just one that they're showing you, and it's not even a complete one here, because there's the esophagus and then the mouth and all the structures related to being able to chew food and swallow it. Anyway, so the digestive system, the organ systems are composed of many different organs. So just as one example, the digestive system, just one example of many organ systems that make up an organism, you've got all these different organs making up that organ system. And each of them have to be functioning perfectly for you to be in a state of balance or well-being. So they have taken the stomach here and sort of shown you that, but certainly you need the liver functioning. They're not showing you the pancreas, but the pancreas is part of the digestive system as well. The large intestines, the small intestines, as I said, the esophagus. So each of them have to be working perfectly. So if we take the stomach, maybe it's the stomach not working well this time. We have to know what the stomach is made of. The stomach is made up of tissues, 2 or more tissue types. So organs are composed of tissues. We are going to learn in histology, the study of tissues, that there are 4 major tissue types. I'm going to give you a couple right now, just as examples. There are 4, but I'm going to give you 2 as an example, and I'll probably give all 4 of them to you, okay? Epithelial. Epithelial, you can Google that and look it up how to spell it. Spelling is important. Start working on it now. Epithelial. Connective is a tissue type. Muscle and nerve. Those are the 4 major tissue types. And I have just told you that all organs, if you took an organ individually, no matter what system you were looking at, all organs are composed of 2 or more of those 4 tissue types. Wait a minute. What are tissues made of? I've already hinted this to you earlier in the lecture, and I told you that tissues are made up of cells. So cells, I want you to know that there are over 200 different types, of human cells. And I'm about to talk to you about something called the cell theory. Okay, so hold on to that. I know you're excited. So hold on to that thought. We're going to come to it in a second. But over 200 different human cell types make up 4 basic tissue types. But what are cells made of? Cells are made of structures, and they have blown this up for you to really magnify this. Structures called organelles, and cells are filled with fluid. So we're going to talk about that when we look at Chapter 3, Cytology, the study of cells. We're going to see some different types of cells, and we're going to go inside of the cell and see these organelles. Let me give you a couple of examples of organelles, just a couple of examples now. You all have heard of nucleus, the nucleus of a cell. You can actually see it on these little illustrations. It's this little round thing inside of the cell. Remember, cells are 3-dimensional. If they weren't, you'd be flat. But they are, and you are not flat. You're 3-dimensional too. So inside of a cell, there's something called a nucleus. That's an organelle. And the one that they have shown you here illustrated, they blew it way up for you so that you could see an illustrated graphic drawing of it. There are mitochondria. So a nucleus and a mitochondria are both examples of organelles that are found inside of cells. But wait a minute, what are organelles made of? What are they made of? They are made of these huge, macro means huge, right? Macromolecules. Macromolecules. The example that they have illustrated here, just one example, is actually an example of a molecule that's found inside the nucleus, the organelle called a nucleus of the cell. This micromolecule is DNA. And I want you, that's an example of a macromolecule. I want you to know exactly what DNA stands for. It stands for deoxyribonucleic acid. 2 words. The first is deoxyribonucleic, and the second word is acid. So that's what DNA stands for. It is a macromolecule. It is said to be the signature molecule of all life. All life. Single cell life, all the way up to the most complex multicellular organisms like animals and plants and fungi. So DNA, a very important molecule, so important that we have an entire chapter based on it. It's called genetics and heredity. So now that was just one example. You have four major, all living things, all living, from the simple bacterial cells to the multicellular complex organisms. There are four major macromolecules. I want you to write those down right now. They are carbohydrates, proteins, lipids, and nucleic acids, of which I just gave you one example. DNA is a nucleic acid. Okay, so, but what are they made of? Y'all are tired of this story right now. I'm almost done. These macromolecules are actually made of smaller molecules, and molecules are made of two or more atoms of elements. Elements being the smallest thing that we know that has unique properties. I'll give you four examples of elements: hydrogen, oxygen, carbon, and nitrogen. They're examples of elements. Actually, all life, from bacterial to complex life, about 98% of you and me and any living form are those four elements. That's crazy to think about. It means that if I were to pull out, like if you're looking at us and you think of yourself as being this whatever, you're really 98% approximately hydrogen, oxygen, carbon, and nitrogen. And if I were to take this organism here and pull out all the hydrogen, pull out all the oxygen, pull out the carbon and the nitrogen, I could sell those four things to a chemistry lab for about $3 to $4. What? What? But how those elements are playing with each other and working together to form these macromolecules that are forming these organelles that are inside of our cells, carrying out very specific functions, is priceless. Now, there are about 20 other lesser elements and some trace elements, about 20 others that are vital for life, but the majority are those four. And understanding how these atoms come together and play or not to make these molecules is called chemistry. And this is why the second chapter of this text is dealing with chemistry. And it's why you can't understand disease in a human unless you understand the basic chemistry. Because disease is not happening at this whole level. It's happening at the molecular and cellular level. And that's where the diagnosis has to come from. We do understand this now. We understand what Hippocrates and Aristotle were trying to get us to understand. And that is that disease, there is a cause and effect. It is not because of your sex or the way you like to have sex or the color of your skin or whatever. It is happening at this level. So we will begin at this level. We can't begin here as much as you all would like to, and I know you would, and actually becomes a little bit easier when we get to the organ systems, but it's because you have gotten this strong foundation from chemistry in understanding how and why atoms of elements come together to play the way they do to form molecules that will come together to form bigger molecules that will have very specific functions inside of organelles that are found inside of our cells. So let's get back to the cell theory. I want you all to know that the cell theory is a scientific theory, and I'm going to talk about that in a second. But the scientific theory that states, the cell theory states these two major premises. And this is me paraphrasing. You can read more about this and whatever, but I do need you all to have the gist of these two premises, and I'm paraphrasing them for you. The cell theory states that, because the scientific theory is a statement explaining a naturally occurring phenomenon, naturally occurring in the natural world. The cell theory states two major things. It states that the cell is the smallest thing that we know that has properties of life. In other words, you think of yourself as being alive, but you're only alive because your cells are alive. And you're only you and acting the way you are because your cells are alive and communicating with each other. If that starts to break down, then there's going to be a problem. If your cells start to not being able to carry out the life functions that they have, there's going to be a domino type of effect. You are the combination of all of your cells carrying out these life processes where they are, as a multi-celled organism, they're communicating with each other. And it needs to be working. Okay. So the first premise is that the cell is alive. And it's the smallest thing that's alive. The second premise is that all life comes from pre-existing cellular life. That's important too for you to get down. All life as we know it on this planet, as we know it, period, comes from pre-existing cellular life. You didn't start off as a multi-celled, trillions of cells, complex organism. You didn't start off that way. You started off as one cell that was alive. Though that one cell came from two cells that were alive that joined and fused, an egg and a sperm. You aren't just what was in the egg, and you aren't just what was in the sperm. Those two living cells came together and fused, fertilization that's called, causing a cell to form known as a zygote, Z-Y-G-O-T-E. I never spell like that for you because I expect you to be able to look up things, but I'll spell that one. You started as one cell called a zygote that had come from two cells that fused in fertilization, and then from that one cell that was the beginning of you, that cell started to divide. And as that cell divided, and divided again, and the 2 divided, and the 4 divided, and the 8 divided, you get what I'm saying? They started to divide. That made you. Now, if we look inside of the cell, we know that there's a nucleus. I had given you the idea that there was a nucleus, and there are these mitochondria, and there are a lot of other organelles too that we'll learn in cytology, our chapter with cytology. We'll learn each of their functions. But when we look inside of the cell and we actually get past this membrane that has a very kind of specialized functions itself and all these different kind of structures itself, nothing inside of the cell is alive. Let me say that again. The cell is animated, is doing work, is reproducing, is had all those characteristics we think of for life, that the cell can divide, can reproduce, that the cell can extract things from its environment and turn them into energy to do work. The cell can make products and release them into the and externally. The cell can actually move things past the cell. It can do all kinds of things. But nothing inside of the cell is alive. Nothing. Organelles are not alive. Molecules are not alive. The fluid, the water, which is mostly water, obviously water isn't alive, is it? But oh my goodness, a cell is alive. Cells can respond to environmental changes. To stay alive, they're going to have to. Cells do carry out very specific instructions that the cell's been given. Cells are doing work 24/7. You get to rest as an organism, but your cells never do. Your cells are 24/7 carrying out functions that are specific to keep you in a state of well-being or homeostasis. So the cell theory is a scientific theory which explains, because that's what scientific theories do. They explain naturally occurring phenomena, and the two major things they're explaining is that the cell is the smallest thing that we know that has properties of life, that's alive, and that all life comes from pre-existing cellular life. You know, you had parents, and your parents had parents, and their parents had parents, and their parents had parents, and we could go back and back and back and back. I also want you to know, I had given you Hippocrates, but now I'm going to give you another person's name, and his name is Louis Pasteur. Louis Pasteur, don't think I have his, I don't know, I don't have his name here, but I want you to write it down. Louis Pasteur is the scientist who was given credit for, finally, after 400 years of scientific research, took 400 years of scientists, some of them spending their entire lives trying to figure out life on this planet and how life came about and what life is and what we define as life processes and where this actually starts. It took about 400 years of research and data and supportive evidence before finally around 1900, I know a lot of you think that was a long time ago, 120 years, it really wasn't. A blink of the eye in geological time. About 1900, Louis Pasteur finally is the one given credit for his research that gave us the cell theory. Louis Pasteur is also known, and I think it's important for you to know this, he's also known as the father of microbiology. Microbiology is one word, and it means microscopic life, the study of microscopic life. So Louis Pasteur, and I do want you to know. Now I want to talk to you about the scientific theory because I've been sort of, stressing this, that you understand this. And I do need for you to understand what a scientific theory, how we actually get to this. And it's so very important that you do understand that it is a statement, it is going to be a statement that actually explains something in nature. Science is limited to naturally occurring things. You can't use beliefs and feel good and oh, that makes me uncomfortable to come into a science class. You can't. Science is all about research, data, using evidentiary evidence to and facts and basis to come up with answers about the natural world. Now, in the same respect, scientists shouldn't be talking about beliefs because there are there are people who everybody has certain beliefs, but beliefs about things that can't be tested, that can't be proven, can't be shown, that aren't about the natural world, can't be brought into science because that would form a bias. So certainly scientists, because people are human, the scientists are humans, have beliefs, but they have to be checked at the door, because otherwise that's going to lead to prejudice and bias in the experimental design. I want you to know that to get to a scientific theory, as I just explained with the cell theory, it took hundreds of years of many scientists doing years and years and repeated research and data to come up with answers sometimes. Now, we have come a long way, and we're kind of in an explosion right now because of all that we can see and get to at a actually at a molecular level. We've got microscopes now that we can see actually at a nano level. So very, very small molecules. Louis Pasteur couldn't even see that 120 years ago. So even in that limited field that he was able to come up with certain things. We're going to learn about some scientists that didn't have all of the things that we have today, but from their observational skills, they were able to deduct certain aspects of what was happening in the natural world that actually led them to have scientific theories. And their theories are spot on even today, and are continuing to be supported by the evidence. Again, in science, you never stop learning, and there has to be a commitment to continual study, even about things that we think we have a good handle on. They'll continue to be researched, and data will continue to be collected. And if new evidence comes to play that would make us have to throw something out that we had thought was the explanation for an event, then you have to do it in science. But if what I'm telling you all is, if you hear that something is a scientific theory, that word theory outside of science means that, oh, somebody has an idea. I have a theory about why she's cheating on him or he's cheating on her. That's not how it's used in science. That's not. A scientific theory is the most exalted explanation by all of the experts in the field to date, using repeated, exhaustive research. Now, I do want, when I talk about research, I want you all to know about the scientific method, and I do want that's used in that research. It's still the best thing that we have right now. It's the scientific method. So I want you, and why I'll explain as we go along, but I want you to know the steps of the scientific method. There are 5. I don't know what you learned before, but there are 5, and you need to know them in order because they're common sense. The first is an observation. The first step of the scientific method is that an observation is made. From the observation, the second thing that happens is a question is formed. Somebody observes something and then asks a question about why that is the way it is. The third step is called a hypothesis, but I want you to know what that simply means. A hypothesis is a statement that answered the question. The fourth is a test, set up some test to test the hypothesis. And the fifth and final step of the scientific method is actually forming a conclusion on those test results, meaning the fifth step is a conclusion about whether you're going to accept or reject your hypothesis. Now, I'm going to give you a scenario that is a little silly, but not really. So I want you to realize that the scientific method is used to answer important questions about the natural world, but you use it every day. You use it dozens of times every day. You open your eyes in the morning, and as soon as you open your eyes, you observe the clock. And you note what time it is, maybe. From that, you ask a question. Why did I oversleep? You answer it because we're human. That's the third step. You answer it. A hypothesis. You say, I overslept because my significant other turned off my alarm. The next thing you do is test that. You can test it by asking them, whatever. You could test it by asking them if they did it, or checking to see if it was turned off and you knew you didn't do it. And then you make a conclusion of whether you accept the hypothesis or not. You make a conclusion, oh, I never turned it on, so my bad, I'm rejecting my hypothesis. Or yes, I asked them and they said they turned it off because they didn't want to hear it go off, and so I accept my hypothesis. The scientific method is logical. It is a logical series of steps about an observation and then trying to answer the question that was generated from the observation. The beautiful thing about science is it never matters if you accept or reject the hypothesis. There is no value based on it because you learned either way. The perfect example there is Edison coming up with the light bulb, right? Supposedly he tested a couple of thousand different materials to try to get to that something that would send that current, and they didn't, so he kept having to throw them out. And someone had actually asked him, didn't you despair over getting so many wrong? He said, no, I learned from each of them. So I learned what didn't work, and that was progress. So rejecting the hypothesis is still learning. So anyway, 5 steps, know them. Observation, a question, a hypothesis, which is a statement that answered that question, testing the hypothesis, and then concluding whether you can accept or reject the hypothesis. That's the scientific method. Now, no one really cares about whether you overslept or whether you ran out of gas and didn't know that you didn't have any gas in the car or whatever. But we do care about things like medications, about coming up with understanding how disease processes happen. In medicine, I'm just talking about in medicine now. Let's be even before medicine though. Let's go back to the idea about our solar system. I don't know if you guys have heard of Copernicus and Galileo, but Copernicus is the one who first, from all of his studies, all of his research, all of his findings, he said that the sun was not revolving around the earth, that the earth was actually revolving around the sun. Now, Copernicus had lots of things that was showing that from his data, but that was such heresy at the time because, man was considered the center of everything, and certainly man was controlling the earth, and everything was revolving around man. And to say otherwise was kind of heresy, was kind of going against some religious things. And so, this was a very controversial thing to be talking about. Now, nobody cared as much about Copernicus as they did Galileo. Galileo was actually one of the most esteemed people in Italy at the time, and he was older, and he was revered and honored, and people loved Galileo. But Galileo started looking at Copernicus's work and started doing his own research, and everything he came up with supported the idea that the earth is actually revolving around the sun. And so he had to state it, and he had to put that out there, and he had to show what he had learned, and he had to say that Copernicus is right. For saying that, they wanted to he went from being the top elder in his society to many of them wanted to kill him. And they discussed it, and it's probably amazing he didn't get put to death, because many did get put to death for that type of thing. Some people got burned at the stake for that. But instead, they decided to keep him under house arrest the last decade of his life. So he was under house arrest. So some of these things, again, though, in science, if people could keep their beliefs and stuff separate from that, it would be great. But it's not always so easy to do, is it? Because a lot of times we as humans want to feel warm and cozy, and want to feel safe, and want to feel, less afraid of things. So it's sometimes really hard to do that. And it's still really hard today for that. It's still continuing. We see it really now in our society. It's pretty amazing in 2021 that we're actually seeing still so much of this. But anyway, so getting back to the scientific method and things about the natural world, which is limited to not beliefs, which are fine and dandy and okay, whatever, but the natural world. To have faith in the results, and this is why it can take sometimes so long to actually come up with the statements that explain the natural event, which is a scientific theory. It's going to take a lot of things. It's going to take huge sample sizes. I could come up with a drug to say, hey, this caffeine can cure cancer, my coffee can cure cancer, because I think it happened on me. Nobody's going to believe it unless you have huge sample sizes. And then not only huge sample sizes, but you also have to have built-in controls, and not usually just one blind control, but double blind controls, where the people don't know if they're and I'm just going to use medicine at this particular time, but people don't know whether they're getting the medicine, and the researchers don't. Everything's been coded, because even though researchers would like to think that they don't have a bias, they may be subconsciously choosing certain members of the control group versus the ones that are going to be actually the ones tested. So to take that out, even the researchers have mixed it up and blinded the results even from themselves until the very end. So control groups where you have blind studies and even double blind studies to try to remove bias, because we are human. Now, the psychosomatic effects are so real. This is so real. So if you don't believe, this is just a proven thing that the power of the mind is amazing. And we definitely know that. And this is why, we understand that positive thinking can have a real positive effect on outcomes and the state of mind that someone's in. But we have to try to remove that from experimental design. So as I said, the experimenter themselves sometimes will be blinded from the studies. And then because of the numbers, the sample size being huge, at the end of the study, when we're making the conclusions from the test results, we can look at those numbers and have the numbers speak to us. Now, it is very important that we understand probability and significance, of these numbers. So not only do we have the researchers that are carrying out the studies and that are doing this, but then we have people that know how to look at those numbers and figure out if there's actually significant data that's being collected that would support a certain statement of whether we accept or reject the hypothesis when we're forming those conclusions. Another huge thing is that in science, we have peer review. There's no scientific theory statement that's been made about a natural event that hasn't been peer reviewed exhaustively and is continuing to get peer reviewed. And what that means is that experts in the field are repeating the scientific method that you went through, and they're getting the same results. And those peers, as a saying goes, have no dog in the fight. That's what I mean to think about dog fighting. Can you believe people ever did that? Anyway, but they don't. They're not, these peers that are getting the same results are not getting any financial gain. They're not getting any professional fame. They're not getting anything for repeating all the things and getting their results. They're doing it for science to see. And if they do get the same results and are verifying it, then that supports that final statement that would be made for that particular scientific theory. So this ensures the honesty, the objectivity, and the quality. What it really ensures is the confidence in the statement. that statement, that scientific theory. It's ensuring the confidence, especially since we know that it all continues to get tested in science. If there's any scientist who is going to take a stance that they can't continue to learn, then they're not a true scientist, because the learning is ongoing. It is, and research is ongoing all the time. There are some scientists that have spent their entire decades of their career, 4, 5, even 6 decades of their career, just studying one protein. One protein. And they're continuing to study. So we understand that. So, and we think about facts versus law of nature, all of these, and the scientific method, they are all used, they must all be used to come together to form that scientific theory. So, as I said, it is very important that you understand that when you hear something as a scientific theory, that is completely different than using the term theory outside of actual science. And that includes outside of actual, like social science. It's different than natural sciences. It's completely different. All right. So with that being said, I have just given you one theory, the cell theory, that states, the smallest unit of life is a cell, and that all life comes from pre-existing cellular life. Another theory that you often hear about is the theory of evolution, right? I want to let you all know that, again, I sometimes have a hard time thinking that it's 2021. There are some schools that won't allow scientific theories to be taught, and there are even some universities that won't allow these because it's threatening to them. So, I don't know why, but anyway, I think it's because of the misinformation and the myths associated with what a scientific theory is, and then also for some of these individual theories, what they actually state. So I think most of you all have probably heard of Charles Darwin, and you would want to know about Charles Darwin, and this is roughly about the time that he was actually coming up with some of this, so you can see about when he was living in the 1800s. It's a fascinating history, really, his history, his family history, and also his research history. But the theory of evolution, I want you to write down what it actually states. Okay, you've all heard of the survival of the fittest and that sort of thing, but that's not really what it actually states. So, kind of it is in a nutshell, but I want to give you another one. The theory of evolution simply states it's not about people, it's not about individuals. It isn't. It isn't about that. It simply states and explains how and why the percentage, so we're talking about a number here, percentage of traits. The theory of evolution states that the percentage of traits, that means another word for trait is characteristics, properties, the percentage of traits or characteristics within a population will change over time. Now, I define trait as another thing to say. You could say characteristics. So the percentage of characteristics you see within a population. So now I need to define population. A population is defined as all the individuals of one species that are located in a particular area. I'm going to say that again. A population are all the individuals combined, all the individuals of one species, within a certain area, and the theory of evolution simply states that the percentage of traits you find within that population is going to change over time. So I want you to think about your ponds. If you've ever had a pond, I grew up on a farm, so we had ponds. But I want you to think about a pond that you know, because it's a small body of water that you can really visualize. In that pond, there are species, there are populations of fish. And if you had carp, maybe you had like little bass or carp, but let's say bass. So you have some bass there. I don't know what you have in the pond. So we're just talking about the bass. Okay, let's just choose bass. This is an example. This is one example of really too numerous to count. But anyway, let's just one example. In that pond, over time, are the percentage of traits within the bass going to stay the same? Are they going to also, are the percentage that you find of traits and those bass going to stay the same for you know, dozens of years? No. You might not have even thought about the traits that bass have, but bass have traits that are attributed to them. They're not all identical. They may look identical to you, but they don't look identical to each other. You know, they don't. So there's a percentage of traits you find. But what would happen if all of a sudden the pond resources started to change? There's something called eutrophication that happens in ponds where they start to get silty and they start to get filled in. And, you know, pollutants can happen, you know, with runoff from farm, you know, stuff that you put on your farm, the phosphates and all the stuff, fertilizers and stuff. So over time, that pond's changing. The environment's changing for the bass. You don't think it's going to affect the percentage of traits within that bass population? It's going to. Temperature changes can affect the percentage of traits. So as temperatures change over time, that can do it. So the theory of evolution simply states that. We can look at, because we like to think it's all about us, but we can look at Homo sapiens. Let's look at us. We're a genus. Genus is Homo. Species is sapiens. We're the only species of that genus. So Homo sapiens, humans. Let's think about humans, let's just say on the planet, okay, because we've realized now we are a global world. We can try to build walls, but too late. Too late. We're a global entity, and we're one race. Homo sapiens. Now, there are percentage of traits that are slightly different in different geographical areas, but that's just because, you know, of the way that we have, time has actually happened with human populations in those land masses. But anyway, so let's think about Homo sapiens. When we think about percentage of traits found within Homo sapiens, do you think that we could just think about like hair color or skin tone or eye color? Are they the same as they were 30,000 years ago? Because there were Homo sapiens on the planet then. Are they the same? No. The percentage of traits you find with them have changed over time. And we know that from fossil evidence and whatnot. Let's think about us as an individual. Did you all know that we have more microorganisms in and on us than we have human cells? The only reason on gross anatomy we look human is because our human cells are larger than the microbes. Our human cells are much larger than microorganisms, but we have way more microorganisms in and on us than we do actual human cells. Okay, so let's think about this. Let's think about the microorganisms. And by the way, you need them. You wouldn't be healthy if you did not have them in balance, in a certain balance. So let's think about the microorganisms that we have in and on us and the ones that we are relying on for our health. Let's think about the ones that are found in our colons, our gut. Our gut microorganisms are actually now being associated with our mental health, our immune health, and so many other aspects of our being. So we need them, and we need them to be there in a certain balance. What if you take an antibiotic? If you take an antibiotic because maybe you have, I don't know, maybe you have a urinary tract infection, or maybe you have a wound infection, or I don't know. You take an antibiotic. When you take an antibiotic, does it affect your microorganisms, the species, the populations of, let's say, E. coli in your gut? You darn well bet it does. And the percentage of traits you find within your one E. coli population will change during that time. They will certainly change during that time. And you know what that is? That's evolution. Every single species that scientists have studied, and we've actually identified the genomes of most, every single species has a known rate of mutational change that's actually just spontaneous and natural, no matter what the environment's doing, that causes the percentage of traits to change over time. That's evolution. That's supporting the theory of evolution. The problem, I think, with these theories, that is the problem, is that people people have misinformation. They think the theory of evolution states that man came from monkeys. No, that's not what the theory of evolution, that's a myth. If man came from monkeys, they'd still be doing it. That's a myth. That makes no sense. So that's not what the theory states. It simply states that, going back, because this again goes back to facts and laws that we know, there's some people who believe, but it's a belief system, that the earth is 6,000 to 10,000 years old. 6,000, because that's roughly when, we think about prehistoric, meaning before men wrote history. So anyway, they think the earth started when man started writing history. So that's the belief system, though. But scientific facts that have been proven state that the earth is about, and y'all should know, 4.6 billion years old. And the evidence all supports that life, though it wasn't life like we know it today, it was single cell simple life, began on this planet 3.8 billion years ago. There was new evidence just a few months ago that has been reported, and it's still being studied, so they're not saying it definitively yet because they're still looking at a lot of data that's coming in, that maybe early life was even a little bit before then. But when we think about 4.6 billion years ago, when the planet was forming, and then 3.8 billion years ago, it's hard to get your head around those numbers, when the earliest life, evidence of the earliest life was on the planet, it was not at all like the life we see today. Actually, all the species that are alive today, if we transplanted ourselves back to 3.8 billion years ago, most would not exist. Within a millisecond, we'd be gone because of the primordial atmospheres and oceans. We wouldn't survive that. But the environment's changed. And as more oxygen was being pumped into the atmosphere, thank you microorganisms, and as the seas changed and the landforms changed and life changed, it evolved. Species, traits, percentage of traits found within the species changed over time, allowing for speciation, which meant a divergence of species. So anyway, that's what scientific theories and science are. The scientific method, I told you the 5 steps. Things that give us confidence in the scientific method are all of these, including peer review and statistical analysis, and then using facts, laws of nature that we see happen. We can come up with an explanatory statement about an event, and that is called a scientific theory. Nothing too scary about that, but that's what that is. So I hope that made sense. I'm hoping I didn't leave out too much stuff, but also to let you know that there's so many things that support the theory of evolution. As I said, we're continuing our study. It's not just something that we come up with a thing and then just stay with it. It's continuing to be studied. But another thing that supports it are the vestigial organs. These are organs that we have in our species of Homo sapiens. We have these structures that no longer have an actual function. And that really makes no sense when we look at other species. Some of these structures have are found in other comparative species, and they continue to have a function, but they have lost a function in us, and this is what is referred to as a vestigial organ, because it's changing over time. The actual function has changed and sometimes been lost. So examples of this are like we actually have muscles for every single hair, even tiny little hairs that you have muscles for them that can cause them to become erect. Now, when you think about goosebumps, that's kind of like what that is, but they're not all working anymore. We actually have muscles that can raise and point our ears forward. Now, some of you can probably wiggle a little bit. Most of us can't even at all do that now, but being able to raise them and lift and point them forward, no, those muscles are not working anymore. We have an appendix, which is a structure that we really don't think has any function, but in comparative species, it's part of the immune system. So, that appendix is often referred to as a vestigial organ. You don't need it. So anyway, I do want you to know we are of the genus Homo and the species is Sapiens. And I do want you to know this hierarchy and how we need to understand this as far as how we're going to go about our approach in learning human anatomy and physiology. So I also, let's see if I want you to, I don't know, if I go past it, remember it's not going to be something that I'm going to tell you about. Now, here's the amazing thing about human anatomy and physiology. We're going to learn what's normal, and we will talk about anomalies, things that fall outside of normal range, anomalies, and we'll talk about diseases and disorders. We'll talk about all of these things, but to understand diseases and disorders and pathology, we have to know what normal would be. And when we talk about normal, there's no judgment based on that. It just means that most of the individuals fall within these sort of standard kind of categories. But here's the most amazing thing, and this is that no two people are exactly alike, not even identical twins. So we'll talk more about identical twins later, but not even really them. So when we think about that no two people are exactly alike, it really becomes a challenge. Think about the surgeons that never know exactly what they're going to get into. They know what should be there when they open somebody up, but they understand that each person is actually kind of unique and different. And so, it really can become, I think it makes it fascinating. So when we look at some of the variations, we do know we'll learn what normal is, but we learn that sometimes people have very abnormal, or they are in what are referred to as anomaly types of situations. So the kidneys, this is what we kind of like to see as a graphic here. We've got little adrenal glands that sit up on top of our kidneys here, ureters going down to the bladder, and then a urethra where urine moves through out to the outside. But some people are born with two kidneys, but might be in very different locations. Some are only born with one kidney, like one regular kind of kidney. Here's a horseshoe-shaped kidney. Some people don't even know that they have these anomalies until, something else happens and they get, scanned or whatnot, and they see that they have those. So a normal heart, and then you've got variations of the heart that can be seen as well. Anyway, so I did talk to you about life, and that the cell is the smallest thing that has properties of life, and that means they can be responsive, there can be movement, they can respond to a stimulus, they can take in a food source and turn it into an energy source to do work. The definition for metabolism, which everyone should have, is that this is all or the sum or the total of really the chemistry that's going on inside the cell. Chemistry, I told you, is how you know what atoms and elements are doing, why and how they come together to play or not. So really, metabolism is this level of work that's going on inside the cell. It's the sum total of all the chemical changes that are happening inside the cell, which is essentially the work that the cell is doing. Not all cells need to have high metabolism at all times. Sometimes certain whole organ systems will be put in kind of not a shutdown, but a slowdown. And then other organ systems are needing to get a boost. And so their metabolism will be upped. Signals will be sent to send them up. And hopefully that kind of makes sense to you. You don't need all your cells at maximum capacity all the time. It wouldn't even be healthy for that to be happening. So characteristics of life. Now I do want to talk to you about homeostasis. And to talk to you about homeostasis, let me see. I am going to do just a quick pause and I will be right back. Okay, so homeostasis, I definitely want to have you all have a working knowledge of what homeostasis is. And I think the best way to do that is with a little drawing. And I'm wondering how, I don't know if I can actually do it on here. This is sad, but that's okay. All right, so I won't be able to do that. I think if there's a way I could actually, no, I don't think so. So, all right. All right, so I just tried to think of a way. I don't think I can. But I want you to know that you could define homeostasis in really one word. You could say balance. You could say well-being, health. What it really means is that the person's body, and they just give you one example here of thousands of things that are going on at any given time. But this one example is this, and this is what I want you to be paying attention to here. This one example is just with temperature control. But what it means is that for every parameter that's being measured, you are measuring, like, not just temperature, you're keeping your pH balanced, you're keeping your oxygen levels balanced, you're keeping your blood glucose levels where they need to be, you're keeping your blood calcium levels where they need to be. There are thousands of things that your body is controlling right this second. Now I'm going to give you an example of just this one because it's the one that your textbook gives you, but then I'm going to give you a quick example of another. So you, though, will know that this is happening. Your body is doing this for thousands of things at any given moment in time. At all given moments in time, they're doing this. So let's just look at temperature. It means that there is a set point that your body would like to be at. And when we think about the temperature, it's really all about your core organs. So your heart, your liver, your internal organs, your brain. That's really what it's about. There is a set temperature. And it's okay that you go up a little bit and that you come down a little bit, as long as you're staying within a certain range that's normal. So there's a normal range for all those examples I just gave you. But when we think about what is setting the set point and what kind of cells, because remember, it's all about your cells, what cells that make up tissues, that make up organs, are setting the set points and making the parameters be what they are, it's the nervous system. So I need you to know it is the nervous system that sets set points and detects when something's getting a little high or getting a little bit low. And you have got to know it is the nervous system. Now, what happens when the nervous system cells actually detect when something's getting a little high? They're going to send a signal to cells that are called effectors that will effect a change. So if the effectors are sent a signal because something's too high, that is going to, the effectors are going to do something that will cause it to go back down. But when the nervous system detects that it's too low, the effectors are going to send a signal that will cause it to come back up. So when the stimulus, whether it's high or whether it's low, is detected and it gets reversed or negated, so when it's high, it's going to be brought down. When it's low, it's going to be brought back up. This is called negative feedback. And you need to know this is how the body mainly stays in a state of well-being, a state of homeostasis, is through negative feedback. That's how it mainly stays. So thousands of parameters. Now, what I'd like to do is to use this, but I want you to forget about temperature here. I want you to forget about temperature. And what I'd like, well, let's continue this for just a second, and then I'm going to give you one other example. So if the nervous system detects that you are too hot, then what is going to happen is your blood vessels are going to relax. They're going to send a signal to effectors in the blood vessel muscles that relax, and blood will come to the extremities, which is going to help you with the sweating process. So you're going to start to sweat, which helps you to cool off. And as you cool off and cool down, you'll get a little bit low. Now, hopefully you won't be so cold that you're shivering. But what happens then is the blood vessels will constrict, the muscles will get a signal from the nervous system, to constrict, sending blood back to your visceral organs. So then you might start your muscles even start to contract, because when you're contracting, you're giving off heat. So these are things to help to regulate temperature. But let's think about blood glucose. Let's think about blood glucose for a second. Blood glucose has to stay within a set normal range called homeostasis. So when we think about blood glucose levels, we can tend to think about 70 milligrams per deciliter as being a low okay. That's okay. And 100, 110 milligrams per deciliter is being an okay high. That's kind of okay, 100. Now, you're going to be vacillating around it though. You're going to be staying within the range, around the set point range. And, when you eat a meal, it's going to go up a little bit higher because almost all meals have carbohydrates, which is glucose in them. But you got to get back down here into normal. Now, the reason that that is so important is that too high of blood glucose levels, and you can write it down as hyperglycemia. Hyperglycemia, one word. Hyper means high. GLYC is glucose. Emia, E-M-I-A, means in the bloodstream. So hyper always means high. Emia always means in the bloodstream, and glyce always means glucose. So that one word, like I said, no such thing as big words. You just break them down and figure out what you're talking about. Hyperglycemia, unchecked, is neurotoxic. What does that mean? It's poison to the nervous system. Oh, wait a minute. What system was keeping thousands of parameters in check? The nervous system. What's the most important system keeping you in balance? The nervous system. Do you want anything that's toxic to it? No. No. And glucose too high for too long is going to end up damaging irreparably nervous system. Now, what about hypoglycemia? And I bet you all know what that means. Hypo means low, glucose in the bloodstream. If it falls below that 70 milligrams per deciliter, what happens then? The cells most affected first, if they don't have enough glucose coming in, is the nervous system. So hypoglycemic is going to depress the nervous system, and it's going to happen quickly. The person might start to feel a little dizzy. They may have problems with their motor ability. They actually often are described as having drunk-like symptoms, like they are inebriated, because their nervous system is depressed. They may not be able to answer your questions. They may be sweating because they have lost homeostasis for many other parameters. So that is deadly too. Where do we need glucose levels? in homeostasis, vacillating back and forth around set point that's all normal and natural. Again, is negative feedback just for blood glucose or just for temperature regulation? No, it's for oxygen levels. Your nervous system, literally, for your bone health, for your digestive system, for all the chemical parameters that are going on in your body, your nervous system, through effector cells like the endocrine and muscles, are keeping you in balance. This is why if you hear of anything that can depress the nervous system or overexcite the nervous system, it's not a good thing, because it would be throwing you out of homeostasis. And not just that one thing. If the nervous system's affected, you can bet it's going to be a lot of parameters that are out. Okay, so homeostasis and negative feedback. Now look, on rare, and you can see some examples of what would have to happen. If we didn't have this happen, when you would stand up every morning, you would pass out because you changed your blood pressure quickly enough. But that doesn't happen. Your body's keeping you in homeostasis. Again, we have to have the nervous system, and then we have to have these effector cells integrating that. Now, on rare occurrences, and this is very rare that this is needed, we have something called positive feedback that happens. And as you can maybe intuitively think, if negative feedback means that the stimulus that was detected will be reversed, so if it's too high, it'll go down, or if it's too low, it will come back up. Positive feedback means the stimulus that's detected will be enhanced. It rarely is needed in the body, and even when it is used, you can only stay in it for short periods of time, but it's certainly needed for childbirth. And this is a perfect example of positive feedback, where this head of the fetus is pushing on the cervix. Cervix means neck, neck of the uterus. Cervical is neck. Cervix means neck of the uterus in this case. So the head is pressing on the cervix. That pressure is sending a signal to the brain to release a hormone. Nervous system detects it, sends a signal to release from the effector cell a hormone called oxytocin that causes this uterus to contract. And it contracts harder each time and longer each time until this head keeps pushing until the head is out, until it's out. And then that oxytocin actually does stay around for a while, because hormones do stay around, until the uterus is back to pre-pregnancy shape and form. But it's that oxytocin that is causing the contraction of the uterus, and it's going to be enhanced. Anybody who's gone through child labor knows that your contractions are, when they start off, they're kind of, you're thinking, oh, I can do this, and they're kind of far apart, but then they get closer together and they become more intense, don't they? Until, that's positive feedback, until the fetus is out and the placenta is out, right? And then well, that's okay, I won't talk about that now. But anyway, oxytocin, a hormone. We have a synthetic version of oxytocin that we can make in the lab. It's called pitocin. Pitocin is sometimes given to women if this process isn't going like we'd like to see it happening, to go ahead and get that fetus delivered. Maybe there's something going on and they want to go ahead and have delivery happen. And so pitocin can be given to help to accelerate that process if needed. We also know that I'll give you a couple of other examples of blood clotting. So if you had a blood vessel that became injured, maybe the blood vessel got cut or something, you need to clot. You need to form a clot so that you don't bleed to death. So a clot formation initially is a positive feedback loop so that the clot can form, breaching, so that it can actually cover the injured part of the vessel so that you don't continue to bleed out. So a blood clot formation is another example of a positive feedback. You can't keep it going forever though, because then the clot would block the vessel, and then blood can't get past that point. So positive feedback until the clot's formed and the vessel's been, the bleeding's been controlled, and then it has to stop. You certainly don't want to stay in labor forever. And it's probably the only thing that gets you through it is that you know it's a short-lived event. It doesn't seem so short when it's happening. But anyway, and then another example that most people can relate to is an orgasm. That's a building kind of event that happens, and then it's the, whatever. So you can think about those three examples. But this is kind of a rare event. Now, I don't want you to misinterpret this slide as saying that all fevers are positive feedback. They really, you don't want them to get into a positive feedback. With a fever, your temperature, your thermostat has changed. So the set point has changed and now gone up. And it's okay to vacillate around that new set point. But the problem, and actually fever is incredibly protective for many different reasons, and we're going to talk about it when we get to that chapter. But unfortunately, every time anybody has a fever now, they want to stop it. They want to stop their fever. But the fact is, they really actually serve a very important function, and they're important. But what isn't good to happen is if you did get in a positive feedback loop. If the fever is going too high, and adults can't withstand as high a fever as infants can, babies can get a much higher fever and be okay, then adults really can't tolerate certain high temperatures. But if it gets in too high and it's not coming back down in that new stadium area region, then it can continue to get high until you are in a positive feedback loop that's going to be fatal. Now, I have never seen 113. I have Fahrenheit. I have seen 108. And I've seen people not make it at 108, actually. And I don't think I've ever seen anybody go above 108 and make it. But your textbook's saying this, so I guess it's happened. Anyway, when we are studying, please keep in mind, it's all about the terminology. So please learn this terminology as we are going. Another really important thing for you all to be doing is to be talking out loud while you're studying. I know that may sound, very foreign to you, but you need to be speaking out loud while you study. They know it works to help you to learn, okay? Pronouncing words, very, very important. Sometimes there are a couple of accepted pronunciations, tomato, tomato, potato, potato, so there are sometimes for words a couple of different accepted pronunciations. Squamous cell, squamous cell, those are both accepted. But you shouldn't be making up sounds, okay? So it's very, kind of important that you do make the effort to, especially in the beginning, to make sure that you are pronouncing things the way that you should. And as I said, it ends up becoming, once you get your words, you get these things built up, it's going to become easier and easier. It really is. I guess I just can't stress this enough. An amazing field. Health sciences is an amazing field to go into. There is no other profession that 100% is always expected, but it's really almost impossible because I've told you that no two people are exactly alike. And even if you have people who have variables that are very similar, maybe they're the same age, maybe they're the same gender, maybe they're the same, I don't know, closely related genetically. They may have the same disease, but their treatment may end up being very different because of how they respond. So it's a challenge, and if you choose to stay in this field, you have to be a lifelong learner. There's new information coming out on a daily basis, things that we're learning about, and it takes a long time for the newest information to get to the bedside, unfortunately longer than we would like to see happening. So it means that the health professionals have to commit to being lifelong learners and willing to take on that. So anyway, thank you all for going into that. You all have in your textbook, the ebook, you have what are called deeper insights. At the index of each chapter, so the index page of each new chapter, you will see a little area that says deeper insights, medical insights. And Usually there are 4 or 5 per chapter, and they're sometimes only a paragraph long, not more than a page long. But you have one on this medical imaging, this non-invasive imaging, and I am going to want you all to make sure that with each chapter you take separate notes on the deeper insights. So please start that right away, keeping those separate notes. As far as this lecture goes, I'm going to go through these kind of quickly, but we do appreciate the medical imaging that's done and how many people that has saved from having open surgeries, right? Exploratory types of surgeries. So x-rays are wonderful. They are flat, flat films that we can see, and certainly like for broken bones and that type of thing is going to be really important. But now we have advanced such that we have computed tomography, and that's what CT stands for. You should know exactly what CT stands for. Some people call it CAT scan, but it stands for computed tomography. This is very low intensity x-rays, so these actually are considered really safe imaging. Sometimes though, when we say that it's non-invasive, it isn't exactly true. You can have CAT scans that don't use inserted dyes, but a lot of times they do want the dyes, and so you can't really call that non-invasive if that's happening. But it can give slices, so that means that you can have these 3-dimensional types of views. Positron emission tomography is injecting radioactively labeled glucose, and so again, that's not quite non-invasive, is it? But what you can see is the activity of the actual cells. I told you that cells have to extract a usable form of energy so that they can do work inside the cells, and you can actually tell from a PET scan which cells are more active than other cells by how this radioactively glucose is being used. As it's being used, it's giving off that radioactivity, and you can see how it lights up in this particular way. These are probably the most sensitive scans that we have for cancer when it's in a very minute, area or tissue. Sometimes the other scans might not show the cells that we know. Cancer cells are greedy, aren't they? They're greedy. They're stealing nutrients and glucose from all their neighboring cells, and their activity is just jacked up. So if you were to look at a CAT scan or MRI, which would be nice, but if you were looking at those, you might miss something that is tiny. But on a PET scan, those little areas would definitely be lit up. So it can be more sensitive sometimes for detecting cancer. A magnetic resonance imaging, MRI, I'm sure you've all heard of that as well. These are really nice. Again, they're really great for soft tissue, but again, giving that 3D type of imaging, which is so much more valuable than just a flat screen. So sometimes they're actually called for as well. And then sonography or ultrasounds you've heard of. These are going to be using these high frequency waves to echo back, and the computer is going to actually change that into an image for you. And now they have become very, very useful as far as prenatal care and diagnostic for diagnostic purposes. So ultrasounds are really valuable as well. So this actually concludes this whole thing on medical imaging is a deeper insight. You took some little notes, brief notes right here, but I would expect you to also maybe look through that, definitely to look through that on from your ebook notes. And there are a few other deeper insights from chapter 1. I know there's one on mystical organs. There's one on what a characteristic of life, like how life can respond to stimuli in the environment where non-living things can't. So there are a few that you have. I think there are 4 or 5 in chapter 1. Take some notes on them, okay? So this concludes our introduction, and what it means is on our next little series of lecture, we're going to be jumping into chemistry.